Hello everyone, welcome back to my channel Anytime Medicine, and today we are going to learn about glycolysis. But before we start, subscribe to my channel so you cannot miss my future videos. So body need energy to function, and that energy comes from ATP, and ATP generates from glycolysis. That's why glycolysis happens in all cells of human body. It occurs in cytoplasm of cell, and it will convert glucose into pyruvate. In that process it will generate ATP and NADH, and that NADH will go to electron transport chain that we are going to talk about in my future videos, and it will generate ATP in electron transport chain. So first thing we are going to learn about NADH. NADH is nicotinamide adenine dinucleotide. It carries electron. As you can see in the picture, that NAD plus will accept electron. This process is called reduction process. In the oxidation process, NADH will donate electron. The donation of electron occurs in electron transport chain, and in the process it will generate ATP. Here you can see the whole process of glycolysis. We will go through each process of glycolysis step by step. First we are going to talk about is hexokinase. So hexokinase is found in almost any part of body tissue. It is inhibited by glucose 6-phosphate. As you can see in a picture, that glucose is converting into glucose 6-phosphate by hexokinase, and it will use one ATP. You have to remember that this process of glycolysis will use one ATP. If there is high level of glucose 6-phosphate in the process, it will inhibit the hexokinase, so this process will be stopped. It has no effect of insulin, and it has high affinity. It means that even if there is a low level of glucose in the tissue, it will work as fine. But it has low capacity. It means that it cannot be worked in the high level of velocity for a long time of period. That's why there will be low velocity maximus and low constant Km. Another enzyme for the same process is glucokinase, which is found in liver and pancreas, and it is not inhibited by glucose 6-phosphate, and it is induced by insulin by promoting the transcription of this enzyme, and inhibited by fructose 6-phosphate. As you can see in the picture, that fructose 6-phosphate is an isomer of glucose 6-phosphate, but it can be overcome by high level of glucose. It has high capacity and low affinity, unlike hexokinase, which means that it can work at high level of velocity for a very long time, but it will need lots of glucose in order to start working. So here is the conclusion of last two slides. As you can see in a graph, that in blue there is hexokinase, which has lower Km and higher affinity. It means that if there is a low level of glucose concentration, it will work in a high level of velocity, which is maximum velocity. In glucokinase there is a higher maximum velocity than the hexokinase, and it will work in a high level of glucose concentration, which means it has higher Km and lower affinity. Now we'll learn about difference between hexokinase and glucokinase in a different level of blood sugar. In low blood sugar, hexokinase will work because there will be no inhibition of glucose 6-phosphate, and there will be inactive glucokinase. And what hexokinase will do, it will deliver the glucose into the tissue. In high blood sugar level, hexokinase will be inactive and glucokinase will work because there is high insulin level and high glucose level. What will do, it will store the glucose as glycogen in the liver. There is a regulatory protein, which is glucokinase regulatory protein, which controls the activity of glucokinase by attaching itself to the glucokinase. It will translocate the glucokinase into nucleus, and by doing that it will inactivate the glucokinase enzyme. Fructose 6-phosphate will induce this process of GKRP binding with glucokinase. Because of that, there will be low level of glucokinase in the cytosol, and there will be low level of glycolysis process. In contrast to that, glucose will compete with GKRP for GK binding, and there will be high level of glucokinase in the cytosol, and it will induce the process of glycolysis in the cell. What if we get deficient to glucokinase? If we get deficient to glucokinase, we cannot do the glycolysis process, and because of that there will be high level of glucose in the blood, and that will result in hyperglycemia. In this disease, pancreas become less sensitive to glucose, and this is exacerbated in the pregnancy. There is disease which maturity onset diabetes of young, which is monogenic. It means that there will be mutation in only one gene which causes diabetes, which is glucokinase. In this disease, there will be mild non-progressive hyperglycemia, and this often worsened by pregnancy-induced insulin resistance. So in this process, we are going to see the enzyme phosphofructokinase. There is two phosphofructokinase enzymes that we are going to see in next slides. First we'll see is phosphofructokinase 1, which is a rate limiting step of this glycolysis process. This is irreversible, so we cannot go back to this process, and glucose will definitely go to the glycolysis after this step, and this cannot go to HM patient and glycogen synthesis. This is a usage of second ATP. Remember that we use second ATP in this process. Phosphofructokinase 1 inhibited by citrate in ATP. Citrate is a component of TCA cycle that we are going to see in my future videos. If there is a lots of citrate in ATP, it will inhibit the phosphofructokinase 1 enzyme, and it will induce by AMP and fructose 2,6-bisphosphate that we are going to see in next slides. Fructose 2,6-bisphosphate at a high level induce the glycolysis process. As you can see in a picture that fructose 2,6-bisphosphate will induce the phosphofructokinase 1 enzyme, and it will favors the glycolysis process, and it will inhibit the fructose bisphosphatase 1 enzyme and stop the gluconeogenesis process. If it get low, it will inhibit the glycolysis process and start doing gluconeogenesis process, as it cannot stop fructose bisphosphatase 1 enzyme. So fructose 2,6-bisphosphate is synthesized from fructose 6-phosphate itself, and it will need phosphofructokinase 2 enzyme. It will convert back into the fructose 6-phosphate with the help of fructose bisphosphatase 2 enzyme, and we'll see that how insulin and glucagon will affect them. So now we'll see how insulin and glucagon will affect those enzymes. Insulin will dephosphorylate those enzymes and will convert fructose 6-phosphate into fructose 2,6-bisphosphate, which means if there is a high level of fructose 2,6-bisphosphate, it will favor the glycolysis by inducing the phosphofructokinase 1 enzyme that we have seen already in previous slides. This happens in fed state when there is a lots of glucose around in our body. Glucagon will phosphorylate those enzymes and will convert fructose 2,6-bisphosphate into fructose 6-phosphate, which means there will be low level of fructose 2,6-bisphosphate into the body. It will favor the gluconeogenesis because it cannot inhibit the fructose bisphosphatase 2 enzyme, and which happens in fast state when there is a low level of glucose and when body need glucose. So now we'll see the further process of glycolysis. Fructose 1,6-bisphosphate converts into two molecules of GAP, which is glyceraldehyde 3-phosphate. This is a reversible process, which means that it can go back to gluconeogenesis. As you can see in the picture, the fructose 1,6-bisphosphate converting into one molecule of dihydroxyacetone phosphate and one molecule of glyceraldehyde 3-phosphate. This dihydroxyacetyl phosphate will ultimately convert into glyceraldehyde 3-phosphate, which means there will be two molecules of GAP. So further in a glycolysis, that one GAP molecule will go to the energy section of glycolysis. That 1-GAP molecule will convert into 1,3-biphosphoglycerate, and that 1,3-biphosphoglycerate will convert into 3-phosphoglycerate. In this process, 1 ATP come out as a product. 3-phosphoglycerate will convert into 2-phosphoglycerate and phosphoenol pyruvate. Phosphoenol pyruvate will convert into pyruvate with the help of pyruvate kinase. In this process, there is 1 ATP come out too, which means there will be 2 ATP from 1 GAP molecule, but we have 2 GAP molecule, which means there will be 4 ATP at the end of the glycolysis process. Remember that the end of the glycolysis process we will get net 2 ATP because we have used 2 ATP in the early process of the glycolysis. So now we'll talk a little bit about pyruvate kinase enzyme. Phosphoenol pyruvate will convert into pyruvate with the help of pyruvate kinase. In this step, we'll get 1 ATP. This is a non-reversible step, and which is inhibited by ATP and alanine and activated by fructose 1,6-bisphosphate, which is an early step of glycolysis process. So how glucagon and epinephrine will affect this enzyme, which will phosphorylate this enzyme and inactivate it. which will slows down the glycolysis process and favors the gluconeogenesis process. At the end, pyruvate will go to the TCA cycle that we are going to talk about in my future videos. So thank you everyone for watching this video. I hope you like it. If you like it, then hit the like button and share it with your friends, and don't forget to subscribe my channel, and please comment down below that if you need any change in my further videos. Thank you.